Okay, so this one we need to talk about energy. When we start out talking about energy, we're going to talk about enzymes a little bit too. First of all, we need to talk about energy and how cells use energy. Cells get energy in many forms, and they have different mechanisms of converting the energy from one to another. The ultimate source of energy from life comes from the sun. That's where we get almost all of our energy. We get all of our energy from the sun, and almost all living things get theirs from the sun too. There are some that get them from other chemical ways, but mostly the sun. First things before we talk about energy, we need to know the difference between matter and energy. Matter is anything that takes up mass, or takes up space and has mass. You, us, we're all made out of matter. Energy is the capacity to do work. To be able to move something, make it move, is doing work. And it's usually expressed in kilojoules or kilocalories. Those are the calories we eat, kilocalories. And that's what energy is. Let's look at energy. Potential energy. Potential energy is the capacity to do work. It doesn't mean you're not doing it. You just have the ability to do it. Standing at the top of a building or the top of a cliff, you have a lot of potential energy, because as soon as you step off that cliff, you're going to start falling really fast, and you're going to expend a lot of energy. It's going to change to kinetic energy, which is the motion energy. So this one is the capacity to do work. You're not doing it yet, but you have the capacity to it. Kinetic energy is the energy of motion, and that's when work is performed. So that's when, if you have watermelon up on the top of the building, that watermelon has a lot of potential energy because it's really high. As soon as you let go of it, it gets a lot of kinetic energy because it's going to be moving really fast. Chemical energy is a type of potential energy. This is very important. It's not kinetic energy, it is potential energy. Right? And that's the kind of energy that our bodies and all our cells use, and it is stored in food molecules, and we get it from food molecules. Now it changes to potential energy. It is chemical energy. It changes to potential energy, and it becomes mechanical energy when we use it in muscles or to metabolism. We've talked about this in the very first chapter. That's all the chemical reactions that take place in our body and in a cell or in an organism. Lots and lots of reactions. There's two different types. We didn't really talk about this before. There's anabolism. That is pretty much when you're putting stuff together. Anabolism is taking small molecules and making them into big molecules. So this is small to big. Catabolism is digesting or breaking things down. That's taking big molecules and changing them into small ones. So you have to break them down to get that to do that. There's two different types of reactions we want to look at. There's exergonic reactions, which are ones that release energy. Energy is released from them. We'll look at these pictures later. An activation energy is the amount of energy that is required to get a reaction to take place. So if it's exergonic, it's going to release energy. You still need to add energy to it to get it to go, but once it goes, it's going to release more energy than you put into it. So it releases energy, but you still have to put energy in it to go. Here's the picture of it. You can see if this is the energy right here of the reactants. They haven't reacted yet. They're not going to until you add this much energy to them. Once you add this much energy to them, they're going to react, and they're going to go, and then they're going to release this much energy. This much energy right here is how much is released. That's why it's called exergonic, because it releases energy. Endergonic is the exact opposite. That one is going to absorb energy or take in energy. Now it still has to have activation energy, which is kind of interesting. So if it starts out here, here's where the reactants are, here's the energy they have. You have to add a whole bunch of energy to get them to take place. They have to get all the way up to here for them to react. Once they do, they're going to react, and they're going to stay right here. They're going to have a much higher energy level from here to here than the reactants did. And so they had to absorb energy because they have more energy than they started with. So that's randomly moving particles diffuse down their concentration gradient. We know that. We've talked about that one lots and lots of times. What that does is it decreases the amount of energy that they have. The best way to think about it is your room, your bedroom. It doesn't want to be cleaned up. You don't want to clean it. It takes a lot of energy to keep your room organized and neat. If you didn't do any cleaning, didn't spend any energy cleaning your room, it would probably be messy all the time. And that's the way these particles want. It takes a lot of energy. If we have a membrane over here and we have all the particles over here, all these particles over here, it takes a lot of energy to keep them over there. They want to spread over here. They want to be nice and evenly spread out. That's the way they want to be. So we got to keep energy, adding energy to keep them over here, right? Just like your room. And that's exactly what happens with a concentration gradient. So let's talk about energy, ATP, chemical that we talked about before that stores the energy. It's the energy currency of the cell is what the term is usually used for that. It's ATP. It is a nucleotide. ATP is a nucleotide. We learned about that when we were talking about nucleic acids. Nucleic acids, and I'll show you a picture in a minute. When we break apart this ATP, it yields ADP and some phosphate, which is the P. So really, there's 3 phosphates. That's why it's triphosphate. It breaks down. We end up with a 2-phosphate plus a single phosphate. That should be a very simple mathematical equation for you guys to figure out. But this is what happens. So here's an ATP. Here's those 3 phosphates. This part right here gives it its name, the adenine and the ribose. So it makes it a nucleic acid. This right here, these phosphate groups, is where the energy is. There's tons and tons of energy right there, and right there, and right there. All those bonds right there have tons and tons of energy, and when those phosphates are released, that energy gets released also. So that's where all the energy is in these ATP molecules. So here's that. Was the opposite of the reaction I was talking about. So if you take ADP, which is adenosine diphosphate with 2 phosphates, add a phosphate to it with some energy, we get ATP. So when we go the other way, we take ATP, energy comes out, the phosphate comes out, energy comes out, phosphate comes out, and we end up with ADP if we go that direction. And then you also know about catabolic and anabolic reactions. So if we're going from smaller to bigger, is that a catabolic or anabolic? You better look it up and see. You should know that one. Here's the reaction of how it's made. You don't need to be able to draw all this stuff. You just need to know there's 3 phosphates here. So energy from the sun, our food, goes and puts this phosphate back onto here, and we end up with ATP. This ATP goes and is used in our cell. We take the energy that's built up in this bond right here, this bond right there, and we break it down and we get ADP again. Now, it's very important for our cells to have a certain ratio of ATP to ADP. We need about 10 ATP molecules for every single ADP molecule, and that's the way our body works the best. If we have too much, it doesn't work. So enzymes. We need to talk about enzymes. These are really cool things. These are proteins. We use some other things in there, but they're usually proteins. So catalysts. Catalyst is something that increases the speed of a chemical reaction. So it increases the rate. It makes the reaction take place faster. And that's just what enzymes are. Enzymes are catalysts. Catalase is the most famous one. It's the one that made those bubbles when we did the peroxide lab at the very beginning. Most enzymes are proteins. It's very important to remember that because that's on the quiz. Most of them are proteins, but there are some RNA enzymes, which we'll talk about when we do. Activation energy. We talked about activation energy a little bit earlier. You saw a picture of it. That is the energy that it takes to get the reaction going, right? And is it lowers it. So here's the activation energy. This is the activation energy of this reaction that wants to take place. These are the reactants. These are the products. This activation energy is really high, but when there's an enzyme around, it's only this high. An enzyme lowers the activation energy and makes the reaction take place at a faster speed. So it lowers the activation energy. Very, very important. Lowers the activation. A good model of that is pushing a rock up a hill. You have to push this rock. This dude has to push it. I don't need that. I'm pushing the wrong button. Hang on. There it goes. Pushing it all the way up here. Once it's up here, he doesn't need to push it to make it go down here. It's going to go on its own because it wants to be down here. This rock wants to. It has lots of potential energy up here, right? Because it's not moving very fast. But he's adding energy to it to increase this potential energy. So enzymes, like I said before, speed up reaction rates by lowering the activation energy. If you know that, you think you're going to get like 3 questions right or something on the next test. The next one. Now, this is how they work. This is kind of the cool stuff here now. Now, what happens is the enzyme brings the stuff together that needs to react. Now, if it's one where there's 2 little pieces getting put together, then it'll bring them nice and close together so they react and bond together. If it's something that needs to break it apart, it'll help kind of break up the product. Now, what happens is the stuff that's reacting is called the substrate. That's the stuff up here. And it binds to the enzyme. It actually fits into the enzyme and makes a complex. They call it the enzyme substrate complex. And you'll see that. Not yet. The place that they bind, the place where the substrate binds, is called the active sites. That's very, very important. It's called the active sites. Now, sometimes the active sites, the things that need to fit in the active sites aren't ready to fit in there. And the thing is, the enzyme has to have something else bind first to make the other piece fit, and that's called an induced fit. So it's the binding of a substrate to an enzyme causes a change in the shape of the enzyme and allows something else to bond. So it's an induced fit. It doesn't normally fit unless it's induced, unless something else helps it fit in there. Now the proximity and the orientation of the reactants is what makes the bonds happen faster and lower the activation energy. So where they are and how the reaction take place faster. So here's a couple of models. This is the most famous one. This is the one you're going to read about in your book when you do that, or if you've already done that section. This is the lock and key model. So everybody's seen a key, and keys only work in certain locks usually. And so the same thing with these enzymes. These enzymes are like the locks, and these substrates are like the keys. So only the right one will fit into each one. Once they do, they're in nice close proximity to each other, and they bind together to make a nice product. Now the other one, induced fit, is the one that, look, they don't really look like they should fit in there, but once they're there, yeah, then they do fit. Right, so these ones look like they fit all the time. These ones don't look like they fit. After they're in there, they do the exact same thing. That's putting them close together, and then they react. Because the shape of the active site is closely related to the shape of the substrate, most enzymes are highly specific. That means they don't just catalyze anything. It has to be the right stuff that they put together. So they only do specific types of reactions. Sometimes they need other parts. Sometimes they need another chemical called a cofactor. Now this cofactor comes along, and for us, cofactors are usually just metal ions or different things that are not organic. Remember, organic is things that are carbon-based. Well, these cofactors are not usually carbon-based. I think I have a picture. Here it is. So this enzyme, this right here, won't do anything by itself. That's why it's called an apoenzyme. It doesn't do anything. As soon as this cofactor goes and binds right here, ooh, red's not a good color. As soon as it goes right here, now the enzyme will work because it's a holoenzyme, which means whole enzyme. Right? So a cofactor just makes the thing that's supposed to be the enzyme that won't work, makes it work. A coenzyme is a cofactor, but it's a organic one. And some of the ones that we have are NADH, NADPH, and FADH2. These ones move electrons, and they're going to be really, really important when we get to photosynthesis. And cellular respiration. Other important coenzymes are vitamins. Everybody eats vitamins, and that's why, because they're coenzymes. They help our enzymes do work. Bind first to help the enzyme get to work. Now, there's different things that affect how well an enzyme works, and one of the biggest ones is temperature. There's an optimal temperature that they work at. There's a perfect temperature or a perfect temperature range for them. If the temperature is too low, they don't work. If the temperature is too high, that's really bad, because enzymes are proteins and they can unfold. That's what denature means, unfold. If they get too hot, they unfold and then they don't work at all. Because remember, the shape is very, very important. There are some that are really cool. These things called extremophiles, they can live in really, really hot temperatures and their enzymes still work. So those are pretty cool. And they use those a lot in the work under such high temperatures. Another thing that affects it is the pH. Remember, pH is how acidic or basic stuff is. Remember, it goes from 0 to 7 to 14, where this is the acids. This is the bases, right? Most things for humans are right in this range right here. If it's too acidic or too basic, it also denatures or unfolds the protein, so it doesn't work. There are some exceptions, because in our stomach, we know that our pH is really, 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 really low. So we hope that our stomach enzymes can handle low pH or very acidic conditions, and they can. That's how they're able to work. Here's some examples of graphs you'll see. So for this green enzyme right here, this green enzyme, this one, you can see its pH that it works best at is the highest point. This is rate of reaction, so the higher up here it is, the faster it's working. Well, it works really, really well at pH of 2 or 2.5 about. That means it's probably one that's in our stomachs. This one up here is working at about 6 to 8, right? That means it's probably one that's floating around our body doing stuff. If it's in a pH way down here, this one's not going to work. This one over here, if it's at a pH up here, it's probably not going to work. So it's very important that they work for where. Now, there's different types of ways of messing up enzymes, and it's called inhibition. Inhibition is stopping something from working. So there's reversible inhibition. That one, it kind of weakly blocks the enzyme from working, and it's good because it doesn't permanently do it. It's reversible. So it can temporarily block the enzyme from working, but then it can be reversed and the enzyme can get back to work. There's competitive inhibition. That's where the inhibitor competes for where the substrate binds, right, to the active site. So it's competing, which is why it's competitive. It competes with the substrate for the active site. Non-competitive one is where the inhibitor binds somewhere else on the enzyme. So if this is the enzyme right here and this is the active site, maybe this non-competitive one will bind right here, and it'll cause this to change shape. It'll make it turn into a square or something, so it doesn't work. It'll change the shape of that. So that's competitive will fight for this spot right here. Non-competitive will bind somewhere else and change the shape and make it not. Irreversible could be either one of those. It means that once the inhibitor is bound to that enzyme, it's done. And those are usually what have been used as poisons. Stuff like mercury and lead. Cyanide, the way they used to kill people with cyanide, it binds to the heme group in your blood cell, and it won't let oxygen bind there. Instead, the cyanide will, and then you die. And that's not good. So irreversible means you can't. It's blocked. You're done. So here's some of the better pictures of this. Here's the enzyme right here. Here's the substrate. This is the active site. Remember, active site right there. The substrate goes in, binds there, and it's happy. Now, when there's inhibition, competitive inhibition is going to bind right here to the active site and block the substrate. Irreversible ones are ones that can't be reversed. Once it's done. Non-competitive are ones that bind someplace else, like this. There's another name for that. It's called allosteric. That's a very good name. Actually, you should know that one. Allosteric means in a different site. Allo means different. Steric means site. So that's a little better picture of it. And that's about it. So that should be some really good notes. And Everybody should get A's on this test. All right.